And so with that, I will turn things over to the moderator for today's session, Dr. Åsa Gustafsson, to introduce our speaker. Please enter any questions for the speaker into the Q&A portion of Zoom, and they will be answered at the end of the webinar. Thank you all again. Looking forward to the talk.
Thank you, Ron. So I want to welcome everyone to the JMCC ISHR cardiovascular webinar. I'm Åsa Gustafsson from the University of California in San Diego, and I'm going to be the moderator for today's webinar. And it is my pleasure to introduce today's speaker, Dr. Joanne Garbincius from the Center for Translational Research at Temple University in Philadelphia. Today, Joanne will talk about her important and interesting work that she's been doing on the mitochondrial sodium-calcium exchanger, or NCLX, in the heart that was recently published in JMCC. Joanne received her bachelor's degree in biological sciences from Cornell University in 2010. And after working for a year as a research technician at Loyola University, she went on to complete her PhD in molecular and integrative physiology at the University of Michigan under the mentorship of Dr. Daniel Michel. She completed her PhD studies in 2017, and her PhD work was then focused on the mechanical regulation of striated muscle nitric oxide signaling in muscular dystrophy. So in 2017, she joined Dr. John Elrod's lab as a postdoctoral fellow at Temple University, where her research switched gears a little bit, and now she's focused on mitochondrial calcium dynamics in cardiac physiology and disease. Joanne is clearly a rising star in the field. She's been very productive and published in many different journals, such as PNAS, Circulation Research, as well as the paper in JMCC that we're going to hear about today. She has received several different travel awards to attend conferences, as well as poster presentation awards. She's also been awarded the very competitive postdoctoral fellowship from the NIH. And I really look forward to hearing your talk today about the NCLX in the heart. And Joanne, the floor is yours.
All right, thank you for that very kind introduction, Åsa. Let me see if I can screen share here. Can you see that? Okay. As Åsa mentioned, I'll be talking today about mitochondrial calcium handling in cardiac physiology and disease. And the first part of my talk really focuses on a question that has challenged our field since the initial determination of the gene encoding the mitochondrial calcium uniporter protein, shown over on the left-hand side of this slide. And that question is, what role, if any, does chronic mitochondrial calcium accumulation play in the development and progression of heart failure of non-ischemic etiology? And then in the second part, I'll highlight some of my ongoing studies into how we might be able to target mitochondrial calcium handling therapeutically, the treatment of heart disease, as well as other diseases characterized by aberrant mitocalcium flux, such as neurodegeneration. So the historical basis for these questions goes all the way back to the 1950s and 60s, with the observation that if you take isolated cardiac mitochondria and give them a substrate like succinate, and let them respire at physiological temperatures, they will readily take up calcium that is added to the bath solution, as you can see in this trace here. And so the outer mitochondrial membrane is readily permeable to calcium ions, and the uptake of calcium across the inner membrane into the mitochondrial matrix is driven by the highly negative electrochemical potential across the IMM. And this is established as the electron transport chain pumps protons out of the matrix. Additional work in the 1970s demonstrated that calcium can also be released from the mitochondria in a regulated manner, as shown in these graphs over on the left. So here we can see that calcium release from isolated heart mitochondria can be stimulated by the addition of sodium chloride. And this same phenomenon of sodium-stimulated mitocalcium efflux can even occur to some extent when the respiratory chain is inhibited, indicating that unlike mitocalcium uptake, this process does not strictly require a functioning electron transport chain. And it would take the field another 40 years or so to identify the specific proteins that are actually responsible for mitocalcium uptake and efflux. But what gradually became clear over time was that altered mitochondrial calcium content is a key mechanism that regulates both cellular energetics as well as cell death. And so under normal basal homeostatic conditions depicted here, cytosolic calcium concentration is relatively low, maybe 100 nanomolar, and there's little sort of net accumulation of calcium or net change in calcium content in the mitochondrion. Resting matrix calcium will be about 150 nanomolar. Now, when cytosolic calcium levels start to rise to over about 400 nanomolar, such as in response to a sympathetic stimulus, rapid mitochondrial calcium uptake becomes activated, shown here. And so in some cell types, this rapid mitocalcium uptake can help to buffer changes in cytosolic calcium and thereby shape cytosolic calcium signaling. But what's important to note for our purposes is that in cardiomyocytes, the mitochondria are thought to buffer at most maybe 1% of the cytosolic calcium transient. So under normal conditions, this rapid mitocalcium uptake does not substantially alter cardiomyocytes' cytosolic calcium signals. Rather, the more important effect of rapid mitocalcium uptake in the heart is that it can increase the mitochondrial ATP supply. And it does that by several mechanisms, including stimulation of pyruvate dehydrogenase, several TCA cycle dehydrogenases, and possibly ATP synthase as well. And this effect is important because it allows cardiomyocytes to increase energy production in order to supply an increase in cellular ATP consumption, which, as in the case of inotropic signaling, will also be enhanced by that same elevation in cytosolic calcium. However, in the presence of very large or sustained increases in cytosolic calcium, mitochondria can eventually take up so much calcium that it reaches the threshold for opening of the mitochondrial permeability transition pore, shown here. And this is a large conductance channel that will allow for the entry of solutes and water into the mitochondrial matrix, causing swelling of the mitochondrion, ultimate rupture of the outer mitochondrial membrane, and the initiation of necrotic cell death. And so if this happens in a terminally differentiated tissue like the heart, the tissue will not be able to replace the dying cells, ultimately resulting in organ level dysfunction. And so given these critical roles of mitocalcium handling in both physiology and disease, the field has put a lot of energy into identifying the specific proteins that mediate this mitochondrial calcium exchange. And the findings of many different labs are summarized here on this slide. So we now know that in the heart, rapid mitocalcium uptake in response to an acute elevation in cytosolic calcium concentration is mediated by a large multi-protein channel of about 600 to 800 kilodaltons called the mitochondrial calcium uniporter complex, or MTCU. This complex is formed by the pore-forming subunit, the mitochondrial calcium uniporter protein, or MCU, and the essential regulator, EMRE. Uniporter function can be regulated by gating of this channel by the intermembrane space proteins, MCU-1 and MCU-2, by the incorporation of a dominant negative pore subunit, MCU-B, by interaction with the transmembrane protein, MCU-R1. Now, on the other side of things, sodium-driven mitocalcium extrusion occurs mainly by the mitochondrial sodium-calcium exchanger, or NCLX. And this is an exchanger that exports 1 calcium out of the mitochondrial matrix in exchange for the entry of 3 sodium ions. And it's important to note here that flux through NCLX represents the rate-limiting step in mitochondrial calcium exchange. So even small changes in NCLX function can actually exert substantial effects on matrix calcium accumulation. I'll also note that there are several other pathways for mitochondrial exchange that have been postulated, such as a mitochondrial ryanodine receptor, the calcium hydrogen exchanger, LETM1, and even the mitochondrial permeability transition pore itself, which has been proposed to have a physiological function as a sort of release valve to allow the release of excess matrix calcium. So once these key players in mitocalcium exchange, MCU and NCLX, were identified in the early 2010s, the field started to manipulate them genetically so that we could dissect what precise role mitocalcium exchange was playing in human disease. And as this particular audience is well aware, one disease that is characterized by features that are closely associated with altered mitocalcium homeostasis, such as disrupted cellular energetics and increased cell death, is heart failure. And what's more, there's actually evidence for altered mitochondrial calcium currents, shown here, as well as altered expression of mitocalcium handling genes, including NCLX, MCU-1, and MCU-B, in both human heart failure, as well as animal models of heart failure. So these observations led the field to ask whether these changes might be adaptive or instead maladaptive, and what specific role altered mitochondrial calcium flux might play in the development and progression of heart failure. And now the first generation of studies really focused on the role of mitocalcium exchange in ischemic heart failure, using mouse models of either ischemia reperfusion injury or permanent myocardial infarction. So here, our laboratory in the Mulkinton lab found that with conditional deletion of MCU in the adult mouse cardiomyocytes, leading to blockade of acute mitochondrial calcium uptake, this was sufficient to limit permeability transition and cardiomyocyte death, and ultimately protect contractile function in vivo after cardiac ischemia reperfusion injury. And our lab reached the same conclusion when we tested the effect of inducible adult cardiomyocyte-specific overexpression of NCLX in cardiac function in IR injury models. So here we find that increasing NCLX activity ultimately increase mitocalcium efflux and thus limit mitocalcium accumulation, would ultimately limit cardiomyocyte death and infarct size, and ultimately preserve contractile function following an ischemic insult. So together, these early studies tell us that with acute cellular calcium overload, excessive matrix calcium accumulation is pathogenic and drives cardiomyocyte death and dysfunction. Now, these findings suggest that limiting mitocalcium uptake in the setting of IR injury or immediately following a myocardial infarction would be beneficial. And while there are some caveats to interpreting some of the genetic models of MCU knockouts, particularly if you start to compare studies of constitutive germline knockouts to conditional knockout in the adult heart, this overall conclusion has stood up pretty well. But what has remained much less clear to us, and what has been a more technically challenging question for the field to address, is whether alterations in mitocalcium handling have any role in the development or progression of non-ischemic forms of heart failure. So here we're talking about heart disease that develops more gradually, such as with chronic hypertension or aortic stenosis, or in hypertrophic cardiomyopathies that will chronically increase the workload and stress on the heart. That's as opposed to ischemic heart failure, which is caused by a single precipitating injury like a heart attack. And so the central question that I faced when I joined the mitochondrial field a few years ago was, what role does mitochondrial calcium have in cardiac responses to chronic rather than acute stress? And in this context, it wasn't immediately obvious whether increasing mitocalcium accumulation over a prolonged time when the heart is subjected to increased energetic demand might be beneficial or instead maladaptive. So some prior work suggests that MCU-dependent mitocalcium accumulation is critical for stimulating energetics to increase cardiac contractility on very acute timescales, such as during physiological fight or flight responses. Yet at the same time, there's an idea which has been proposed by Christoph Mack and Brian O'Rourke, which suggests that in end-stage heart failure, you can actually get an increase in cytosolic sodium accumulation. And this might actually drive excessive mitocalcium efflux through NCLX, ultimately starving the mitochondria of calcium, which could then impair mitochondrial energetics and redox balance. And so in this model, mitochondrial calcium accumulation might actually be a good thing for a heart that's already failing. On the other hand, there's some anecdotal evidence that links increased mitochondrial calcium uniporter expression to pathological hypertrophy with either chronic pressure overload due to hypertension or aortic stenosis. And finally, there are some reports in the literature of either no detrimental or no beneficial effects of either constitutive MCU knockout or adult cardiomyocyte-specific MCU knockout when you look at cardiac remodeling and contractile function following pressure overload. So trying to piece all these observations together, we came up with the hypothesis that mitochondrial calcium accumulation would contribute to the heart's adaptation to a chronic increase in the heart's workload for its effect on energetics. That this same calcium signal might also contribute to pathological remodeling and eventual cardiac maladaptation. And we set out to test this hypothesis with two complementary in vivo studies, the first of which compared the effects of cardiomyocyte-specific MCU knockout versus overexpression on the heart's response to chronic beta-adrenergic stimulation. So for this study, we first developed and validated a mouse model of tamoxifen-inducible adult cardiomyocyte-specific transgenic MCU overexpression. So here, we have a construct where MCU is downstream of a lock-stop-LOX cassette. When we cross this to an alpha-myosin heavy chain mercury line, then administer tamoxifen, we can induce a robust MCU overexpression in the cardiac mitochondria. Next, to assess whether this overexpressed MCU protein incorporated appropriately into high-weight molecular uniporter complexes, we took cardiac mitochondria from these MCU transgenic mice, or our Cree controls, and separated them into different molecular weight fractions using size exclusion chromatography. And so as you can see in our mercury controls, we see that most of the MCU in the mitochondria is present in these high molecular weight fractions. The size is shown down here. And these range from about 400 to 800 kilodaltons. And this is quantified in this black trace. In our MCU transgenic mitochondria, overexpressed MCU likewise partitions into these same high molecular weight fractions, and that's shown in this blue trace. So this tells us that this overexpressed MCU does incorporate appropriately into the mitochondrial calcium uniporter complex, probably with the appropriate stoichiometry with the other subunits. Next, to validate that our model was working the way it should, we checked that MCU expression was actually increasing mitocalcium uptake. So for this assay, we isolate adult mouse cardiomyocytes, place them in a little cuvette, and permeabilize the cells with digitonin so that the cytosol essentially becomes continuous with the bath solution that's in the cuvette. We can then monitor the extramitochondrial calcium content using this calcium-sensitive dye, Hela. And then what we do is administer boluses of calcium, as shown here. You see that there's an initial spike in the calcium in the bath solution, then that gradually decays as the calcium is taken up into the mitochondria. And so, as expected, we see that this rate of mitochondrial calcium uptake is increased in our MCU transgenic cardiomyocytes, shown in blue. And consistent with this phenotype, we see that isolated MCU transgenic mitochondria are more likely to undergo swelling and have this drop in absorbance in response to addition of a large bolus of calcium. That tells us that we are successfully increasing net cellular MCU activity, which will then cause mitochondrial permeability transition and swelling in these overexpressor cells. And then finally, to verify that this MCU overexpression is having the expected physiological consequences, we see that MCU overexpression decreases the inhibitory phosphorylation of pyruvate dehydrogenase in cardiomyocytes. And this is consistent with the activation of a matrix calcium-sensitive PDH phosphatase. And finally, this effect of more PDH activation is then associated with an increase in the basal respiration of these cardiomyocytes. Next, we took this MCU overexpressor model alongside mice with tamoxifen-inducible adult cardiomyocyte-specific MCU knockout that the lab had published previously, and subjected them to 2-week infusion with isoproterenol using osmotic mini pumps. Now, when we run this experiment, we see that in our mercury controls, shown in these black bars, there's an increase in contractility over the first few days, and that persists somewhat throughout the course of our experiment. And these mercury control mice also exhibit cardiac hypertrophy by our 2-week endpoint. Now, in contrast, MCU knockout mice, shown in red, really failed to increase cardiac contractility over the 2 weeks of the ISO infusion. This suggests to us that MCU-dependent mitocalcium uptake is indeed required to support an increase in cardiac work output over the course of days to weeks. And this really contrasts with the idea that alternative routes of mitocalcium uptake or alternative mechanisms of metabolic regulation are sufficient to compensate for the lack of MCU over any time frame longer than, say, a minutes-long acute fight or flight response. Interestingly, though, we still see cardiac hypertrophy in these MCU knockout animals, suggesting that MCU itself is not strictly required for these hypertrophic responses to chronic stress. And then when we go ahead and look at our MCU transgenic mice, the early contractile response to isoproterenol is largely preserved at 2 days of ISO, as shown in these blue symbols here. However, with continued stimulation, these animals begin to decompensate, and they show exaggerated hypertrophic remodeling compared to our pre-controls. So this indicates that although MCU-dependent mitocalcium uptake is required for supporting increased contractility, this same signal will ultimately put the heart at risk for dysfunction if it is maintained at too great an extent for too long of a time. Next, to get a handle on the ultimate cause of this exaggerated contractile dysfunction that we see in our MCU-Tg hearts, we examined isolated cardiomyocytes in vitro. And so here we see that our MCU transgenic cardiomyocytes have increased ROS production in response to treatment with a calcium ionophore and amikacin, as well as at basal cytosolic calcium concentrations, which we can sort of uncover if we inhibit respiratory complex 3 with antimycin A. And then we also see an increased sensitivity of these MCU transgenic cardiomyocytes, calcium-induced cell death during ionomycin treatment, quantified here, and a tendency towards increased cell death in response to acute isoproterenol treatment in vitro. And so based on these findings, we hypothesized that in vivo, MCU overexpression could be driving contractile dysfunction during chronic isostimulation because the resulting mitocalcium overload and oxidative stress, of course, would be causing mitochondrial permeability transition and ultimately necrotic death of the cardiomyocytes. So to test this notion, we turned to mice with knockout of the PPIF gene, which encodes cyclophilin D, which is the only genetically confirmed component of the mitochondrial permeability transition pore. So prior work has shown that knockout of cyclophilin D is sufficient to protect mice and mitochondria against mitochondrial calcium overload induced permeability transition. And you can see that on these mitochondrial swelling assays shown here. So this cyclophilin D knockout is in this pink trace, does not swell in response to bolus addition of calcium. What's more, this knockout of cyclophilin D is sufficient to protect cells from necrotic cell death, as indicated by propydium iodide staining in response to different agonists that will increase cytosolic calcium concentration. Our lab has also previously shown that this strategy of using cyclophilin D deletion is protective in vivo in a model of massive mitocalcium overload that we can induce in mice that lack NCLX. And so here, comparing the red and the green groups, we see that cyclophilin D deletion in green improves the survival and protects the contractile function of mice with cardiac NCLX deletion, which is the red group here. So, okay, so we took these cyclophilin D knockout mice and we crossed them to our MCU transgenic model to test whether the deleterious effects of MCU overexpression were mediated through classical mitocalcium overload-induced permeability transition and cardiomyocyte necrosis. Then we took these animals and subjected them to our same 2-week isoproterenol infusion protocol. So here, the question we're asking is whether deletion of cyclophilin D or PPIF will be able to rescue this contractile dysfunction that we see in our MCU-Tg mice that are subjected to iso infusion. So these dark blue bars in the wild type background. And the answer is no. So when we knock out cyclophilin D in our MCU overexpressor mice, here looking at the light blue bars, we're not able to rescue either fractional shortening, lung edema, or cardiac hypertrophy. Next, we asked if this cyclophilin D knockout strategy was actually able to alleviate iso-induced cardiomyocyte necrosis in our transgenic mice, as it has been shown to do in other cellular calcium overload models. And so the way we did this was by injecting our mice with Evans blue dye, which will bind to serum albumin and is normally excluded from cells that are healthy and have intact plasma membranes. However, if you have a cell where the integrity of the plasma membrane becomes compromised, such as in a cell undergoing necrosis, that will be able to enter the cytosol and label the cell. And we can then visualize that as red staining on a fluorescent microscope. So here, just starting with the MCU transgenic on a wild type background, we see that isoproterenol infusion increases cardiomyocyte necrosis, as quantified by this Evans blue dye staining here. And then if we go ahead and knock out cyclophilin D, we had a very surprising result, which was that this deletion of cyclophilin D did not attenuate and actually exacerbated this cardiomyocyte necrosis in our MCU overexpressors, quantified here in the light blue. So this tells us that sustained mitochondrial calcium loading under chronic stress is sufficient to cause cardiomyocyte death, and that this can occur even independent of classical permeability transition. And so this finding hints to us that in these really chronic cellular calcium stress states, the more important role of permeability transition pore may be to aid in mitochondrial calcium efflux and to alleviate mitochondrial calcium overload, rather than functioning as the sole key driver for cardiomyocyte necrosis. And this idea actually fits with what has been observed previously in cyclophilin D null hearts, where there's little impacts on baseline function of the heart. Matrix calcium content is increased, and then these animals are sensitized to having poor contractile performance when they're subjected to a chronic stress paradigm like pressure overload or forced swimming exercise. And so to recap this first study, we concluded that MCU-dependent mitochondrial calcium accumulation is required to support these chronic increases in cardiac workload under sustained sympathetic stimulation, and that alternative mechanisms of either slow mitochondrial calcium uptake or alternative mechanisms of metabolic regulation are not sufficient to support this increased work rate. What's more, our data suggests that mitochondrial calcium accumulation can contribute to pathological hypertrophy under chronic stress, although this effect does not seem to depend specifically on calcium uptake through MCU. And finally, although mitocalcium accumulation through MCU is adaptive in the short term, given enough time, it can predispose to cardiomyocyte death and contractile dysfunction, even independent of classical necrotic cell death pathways. One question that our first study could not fully address is whether any mitochondrial calcium accumulation that could occur independent of MCU, for instance, through some of these alternative pathways that I highlighted earlier, plays any role in non-ischemic heart failure. And in particular, might it have any role in pathological remodeling, which was not alleviated in our MCU knockout model. So we decided to complement our initial study with an alternative approach to test our central question. Here, instead of manipulating mitocalcium uptake through MCU, we next asked how we could affect the progression of pathological remodeling and non-ischemic heart failure if we instead increased the net activity of NCLX, the mitochondrial sodium calcium exchanger. So here, we used a genetic mouse model that our lab made previously, which allows for adult cardiomyocyte-specific overexpression of NCLX. And using this model, we get about a twofold increase in cardiac NCLX expression. And this corresponds to about a 38% increase in the rate of mitochondrial calcium efflux. We then subjected these mice to chronic pressure overload using transverse aortic constriction and followed their cardiac function out for 12 weeks using echocardiography. And so here, we find that NCLX overexpression, shown in this dark blue line, prevented the gradual decline in contractile function that we otherwise see with chronic pressure overload. So just like our MCU study, this result suggests to us that chronic mitochondrial calcium loading eventually becomes deleterious and compromises contractile function. In terms of pathological remodeling, we saw that NCLX overexpression reduced cardiac fibrosis. And so this is consistent with protection from deleterious mitocalcium overload, cardiomyocyte death, and replacement scarring of the myocardium. NCLX overexpression also attenuated cardiac hypertrophy with chronic pressure overload. And we see that whether we look at heart rate to tibial length ratios or cardiomyocyte cross-sectional area. And we are very interested to see this anti-hypertrophic effect of NCLX. But one of our key questions was whether this effect was directly attributable to changes in mitocalcium handling, or rather might just be a secondary consequence of reducing cardiomyocyte death and then alleviating the excessive wall strain that would be placed on the remaining surviving cardiomyocytes. So we went back and ended up looking at an earlier time point, just 2 weeks post-TAC, before we had any evidence of cardiomyocyte death, fibrosis, or contractile dysfunction. Here, we again see that NCLX overexpression attenuates cardiomyocyte hypertrophy even very early in this disease process before we have other signs of pathology. We also saw a similar anti-hypertrophic effect of NCLX overexpression using a pharmacological model of 2-week low-dose angiotensin infusion using osmotic mini-pumps. And what was interesting in these cases was that this early anti-hypertrophic effect of NCLX often occurred despite preserved activation of fetal gene programs that are classically associated with hypertrophy. And this suggests that the hearts are still feeling this stress, and some aspects of the normal response to these stressors are still intact, even despite NCLX overexpression. Yet somehow, when we overexpress NCLX, we're still able to put a brake on cardiomyocyte growth. And finally, to test how robust these protective effects of NCLX overexpression were, we turned to a pharmacological model of extreme neurohormonal stress using a combination of high-dose angiotensin 2 plus phenylephrine. These are both alpha-adrenergic agonists, which will increase intracellular calcium cycling. So here, we saw that the drug infusion caused this patchy fibrosis throughout the myocardium of our control mice, suggesting that it was severe enough to kill the cardiomyocytes. Again, this could be attenuated if we overexpressed NCLX. And once again, we see that NCLX overexpression is attenuating cardiac hypertrophy using this model. So up to this point, all of the data from our 2 studies suggested that sustained mitochondrial calcium loading in these conditions of a chronically increased cardiac workload or chronically increased cytosolic calcium signaling would contribute to hypertrophy, cell death, and contractile dysfunction. And to explore in some more mechanistic detail exactly how increased mitocalcium content was contributing to pathological hypertrophy and how NCLX was able to attenuate it, we next moved to a more reductionist model. So here we're using 48-hour stimulation of neonatal rat ventricular cardiomyocytes with phenylephrine in vitro. So here, using a genetically encoded calcium reporter, we see that adenoviral NCLX expression limits steady-state mitochondrial calcium accumulation during phenylephrine stimulation, even though it doesn't decrease and actually increases steady-state cytosolic calcium content. And as we would expect, with less mitochondrial calcium loading and potentially less stimulation of the TCA cycle, we see that NCLX also limits mitochondrial ROS production during PE treatment. And then it also attenuates phenylephrine-induced increase in cellular oxygen consumption. And so we interpret these data as indicating that NCLX overexpression, which will then limit mitochondrial calcium accumulation during the hypertrophic stress, is essentially putting a brake on mitochondrial oxidative metabolism. And the consequence of this limited catabolic activity is that the cardiomyocyte's anabolic potential ultimately becomes limited as well. So we can see that here, if we measure de novo protein synthesis using puromycin incorporation assays, we quantified in this graph. So where phenylephrine treatment increases the rate of protein synthesis in cells transduced with the control virus, shown in this black trace, this effect is blocked in cells that are transduced with virus encoding NCLX, and there's again shown in dark blue. The ultimate result of this attenuated rate of protein synthesis is that in the course of our full 48-hour experiments, cardiomyocytes treated with phenylephrine will grow less if they overexpress NCLX. So together, these data suggest to us that by limiting mitochondrial calcium accumulation, NCLX is able to limit oxidative catabolism that is normally required to fuel biosynthesis of macromolecules such as proteins that are required for a cardiomyocyte to physically grow. So for instance, the addition of each amino acid to a nascent protein requires ATP to charge the tRNA and energy in the form of GTP to carry out the translation reaction at the level of the ribosome. Another interesting idea here that we have yet to test directly is that if NCLX overexpression is in fact limiting TCA cycle flux during hypertrophic stimulation, that effect may also limit the production of essential building blocks like amino acids, lipids, and nucleic acids, whose synthesis is linked to TCA cycle metabolites. So from this work, we conclude that increasing mitochondrial calcium efflux in cardiomyocytes will mitigate the pathological remodeling of the heart via at least two mechanisms. First, it appears to reduce the likelihood of mitochondrial calcium overload and cardiomyocyte necrosis. And then second, stimulating mitochondrial calcium efflux can attenuate the primary hypertrophic response of the cells. And so this is consistent with a model in which the extent of cardiomyocyte growth is proportional to the amount of time that mitochondrial calcium content is elevated above some threshold level. And so when we increase the capacity for mitochondrial calcium efflux by overexpressing NCLX, we can shift this mitocalcium accumulation curve to the right and ultimately reduce the amount of time that the cells have to grow. This results in less cardiac hypertrophy at any of our experimental endpoints. And so according to this scheme, limiting mitocalcium accumulation in the chronically stressed heart should be largely beneficial, as it will limit a lot of the pathological remodeling that can ultimately compromise contractile performance. And one other thing that I'll note here is that in these chronic heart disease models, PEC, neurohormonal overload, et cetera, NCLX may be a more effective therapeutic target than the mitochondrial calcium uniporter for limiting certain deleterious aspects of continual mitocalcium loading, such as fibrosis and hypertrophy. However, as we finished up these studies and ran some more mice, one other feature became clear. So we were very surprised to see that in our extreme calcium stress model with Ang2 phenylephrine infusion, NCLX overexpression was actually detrimental in the early phase of this drug infusion, shown in this blue line on the survival curve. So we see a significant increase in mortality throughout the first 2 weeks of this Ang2 infusion if we overexpress NCLX. And it turns out this is very similar to an effect that we observe if we overexpress NCLX in mice that have transgenic overexpression of the beta-2A subunit of the L-type calcium channel. So just to refresh everyone's memories here, the beta-2A channel transgenic model leads to constitutive activation of the L-type calcium channel, depicted here. And prior work has shown that beta-2A expression alone can increase cytosolic calcium transients and cardiomyocyte necrosis. And when these beta-2A transgenic mice are subjected to chronic isoproterenol infusion, this massive calcium overload and necrotic phenotype can be exaggerated and even lead to cardiac dysfunction. So when we went ahead and crossed our NCLX overexpressor mice to these beta-2A transgenic mice, and then put them on chronic iso infusion, which induced a massive increase in cytosolic calcium handling. And here we saw that there was this early increase in mortality in the mice with NCLX overexpression. This was specific to the first couple of weeks of the study, and any animals that survived this sort of initial window of vulnerability then seemed to do okay. So we're still not clear why exactly this occurs. But one possibility is that if we really are augmenting mitocalcium efflux in these very extreme calcium stress conditions, we might starve the mitochondria of calcium to the extent that it limits TCA cycle flux and possibly throws off mitochondrial redox balance. This could result in oxidative stress that causes SR calcium leak, leading to cardiac dysfunction on a larger scale and possibly fatal cardiac arrhythmia. Alternatively, if we limit mitocalcium accumulation in the setting of extreme cytosolic calcium overload, it's also possible that we would favor increased plasma membrane sodium calcium exchange, which could then depolarize the plasma membrane and increase the propensity for arrhythmia and sudden death, as we see on these survival curves. So this last set of results cautions us that increasing mitochondrial calcium efflux and limiting mitochondrial calcium accumulation is not always beneficial or even neutral. If this occurs during extreme chronic cellular calcium stress, it can in fact be detrimental. And so this finding really highlights one of the nuances of mitochondrial calcium signaling that we've come to appreciate, which is that both mitochondrial calcium overload and mitochondrial calcium depletion can be deleterious for the heart. So this reinforces the importance of mitochondrial calcium signaling to physiological function, as the function of a pathway, which is critical to a cell, often must be kept in a very tightly controlled window, with either too much or too little activation of that pathway causing dysfunction. And so our therapeutic goals for cardiac pathologies that are characterized by aberrant mitocalcium homeostasis really shouldn't be aiming to completely ablate or completely overactivate the routes of mitochondrial calcium uptake or efflux. A better strategy is probably to tune the functionality of those proteins in order to keep mitochondrial calcium content in a more sort of physiologic range and avoid either overload or depletion. And so for NCLX, this means that we would need to stimulate or inhibit its activity as the particular circumstances of a given disease demand. So in the last few minutes, I'd like to preview some of my ongoing in vitro work that seeks to determine how exactly we can tune NCLX activity to keep mitocalcium in this more physiologic range and treat human disease. So in addition to the work that I've summarized already regarding NCLX and heart failure, there's a growing body of evidence that NCLX may be a good therapeutic target for additional diseases that feature changes in mitocalcium homeostasis, such as Alzheimer's disease and neurodegeneration, as well as cancer. However, a major challenge in achieving this goal of manipulating NCLX therapeutically is that currently very little is known about how the exchanger activity of NCLX is regulated. And it turns out that this question very quickly becomes complicated because NCLX, depicted here, is what we like to term as a proteomic nightmare. It's this 13-pass transmembrane protein localized at the inner mitochondrial membrane, and it's very difficult to isolate, especially because none of the available NCLX antibodies are very good. So the approach that we took was to perform a proximity biotinylation screen. And the idea was that we could identify endogenous proteins that would lie in the vicinity of NCLX and that may regulate its function. And so it turns out that plasma membrane sodium calcium exchangers can be regulated by allosteric interactions with other proteins. So we hypothesized that NCLX might be regulated in this same way. And so the way this proximity biotinylation screen works is that you make a fusion protein with your protein of interest attached to a promiscuous biotin ligase that will biotinylate lysine residues on any proteins that come within about a 10 nanometer radius of the fusion protein. You can then express your fusion protein in your cell type of interest. You add biotin and allow any proximal proteins to become biotinylated, whereas more distal proteins will not be labeled. You can then affinity purify the biotinylated proteins and identify them by mass spectrometry. And so for our screen, we designed a fusion construct with the biotin ligase enzyme, bioID2, attached to the C terminus of NCLX, followed by an HA tag. And so when we express this in AC16 cardiomyocytes, this fusion protein localizes to the mitochondria, as you can see in this HA blot. And then if we isolate mitochondria from cells expressing this construct and subject them to digestion with increasing concentrations of trypsin, we see that the degradation of the fusion protein, again, looking at this HA blot, is somewhat delayed compared to degradation of outer mitochondrial membrane protein TOM20. And some of these cleavage products persist even at our highest concentrations of trypsin, where TOM20 is completely degraded. So this suggests that the C terminus of the fusion protein, which contains this HA tag that we're plotting for, might be localized within the mitochondrial matrix and thereby protected from degradation. So next, we took 3 replicates of samples from AC16 cells or Hela cells that were transfected with either a bio-ID alone construct or NCLX bio-ID fusion protein, and then cultured in the presence of biotin. We took these samples, submitted the proteins from mass spectrometry to identify which proteins were biotinylated. And our results are summarized here. So after taking the biotinylated proteins that were enriched at least 2-fold in NCLX bio-ID compared to bio-ID alone, in at least 2 out of our 3 replicates, we then cross-referenced those proteins to the integrated mitochondrial protein index database. This is a list of all known and predicted mitochondrial proteins. So this got us down to about 100 mitochondrial proteins per cell type that were identified as being proximal to NCLX, and then just 25 mitochondrial proteins that ended up being enriched in both of our cell types. We then prioritized our list of candidate NCLX-proximal proteins for further screening based on several criteria, including whether there was published proteomic evidence for the protein being expressed in mitochondria and tissues of interest, such as the heart and the brain, and whether their expression changed in mouse models of diseases such as heart failure. So this got us down to our top 20 or so candidate NCLX-proximal proteins that we decided to follow up in vitro. So to test the effect of these genes on mitochondrial calcium efflux, we did 24-hour transient transfections in AC16 cells using plasmids encoding each gene of interest, followed by a MYC flag tag. So this is very similar to the mitocalcium flux assay I showed before. So we're taking these cells, putting them in a cuvette, permeabilizing them so the bath solution is continuous with the cytosol. We can then give a bolus of calcium, shown in this black line, and see that there is an increase in the extramitochondrial calcium that then rapidly decays as the calcium enters the mitochondria. We can then treat the cells with an MCU inhibitor, RU360, and this will block further mitocalcium uptake and allow us to monitor efflux of calcium from the mitochondria. So as you can see here, when we do that, mitochondria gradually spit out just about all of the calcium that they took up in response to that first bolus. And then finally, we conclude the experiment by adding FCCP to depolarize the mitochondria and dump any remaining calcium from the matrix. So using this assay, we found that 5 or 6 of our top candidates either inhibit or stimulate mitochondrial calcium efflux. And one of our most interesting hits, which looks like it is inhibiting mitocalcium efflux, shown in this green trace, is depicted here. And we've gone ahead and confirmed that this particular gene localizes to the mitochondria, as you can see with its co-localization with the mitochondrial protein TOM20. Some of my preliminary data suggest that this gene is highly expressed in the mouse heart and skeletal muscle. And we also find that after either TAC or myocardial infarction, both situations of potential mitocalcium overload, cardiac expression of this gene decreases acutely. So if this particular protein does in fact inhibit mitochondrial calcium efflux, as our preliminary data suggest, this could be a natural compensatory mechanism to help limit deleterious mitochondrial calcium accumulation in these disease states. And so we, of course, have a lot of work left to do to confirm whether this gene and our other candidate regulators are indeed acting through NCLX or via other mechanisms. But we're very excited about where this work is headed and its potential to reveal novel mechanisms that regulate mitochondrial calcium exchange. And so to summarize, I've shown you that MCU-dependent mitocalcium uptake is specifically important for supporting not just acute, but also sustained increases in cardiac work during chronic adrenergic stress. Yet, continual mitocalcium loading under chronic stress can predispose to cardiomyocyte dropout and eventually compromise contractile function, even independent classical necrotic signaling pathways. A sustained increase in mitocalcium will also help provide the fuel needed for the heart to hypertrophy in these pathological settings. And yet, at the very onset of extreme stress, mitocalcium accumulation also appears to have a beneficial role because it somehow is mitigating the risk of sudden death. And so together, these findings really reveal that there is no single therapeutic goal regarding mitocalcium handling that's going to be appropriate for all situations. So depending on the type of cardiac stress we're considering, ischemic or non-ischemic, in the particular stage of any one patient's disease, you may want to increase or decrease mitochondrial calcium accumulation. To that end, our work is now focused on understanding the endogenous regulation of NCLX activity, with the expectation that deciphering those mechanisms will be a key to unlocking new strategies for therapeutic manipulation of mitochondrial calcium homeostasis. And so with that, I'd like to thank my mentor, John Elrod, as well as our funding sources, and everyone who supported this work. And I'd be happy to answer any questions. Thank you.
Thank you so much for that beautiful talk. Very interesting. So if you have any questions, type it in the Q&A. I see that we do have a couple of questions, but I wanted to start off by just asking the first question. So all of your experiments have focused on the pathological hypertrophy. So what about physiological hypertrophy? What do you think will happen if you exercise your mice?
Yeah, great question. So we haven't done that particular experiment yet. What I can say is that other models where you manipulate net mitocalcium accumulation, for instance, this is one of the consequences of knocking out cyclophilin D, they tend to see exaggerated, I don't know if you could still even call it physiological hypertrophy in that case, in response to models like forced swimming exercise. So it's probably physiologic for a time, and then it decompensates into being pathological and problematic. So my guess would be that, you know, if I guess our primary data in the isolated cardiomyocytes suggests that, you know, if you were to say boost NCLX activity, you might be able to even put a brake on just the physiological growth of a cardiomyocyte, as well as this pathological state. But again, the balance of doing that without causing more problems is tricky.
Yeah. Okay. Okay, thank you. So let me read the first question from Daniel Johnson. Really nice work and talk, thanks. I was wondering if you also looked at the expression of adrenergic receptors in the MCU knockout animals. Even if there was no changes, did you look at alterations in location of these receptors or changes in the different mitochondria population, that is subsarcolemmal, interfibral? Maybe this would also be interesting to look at in the NCLX, B2 transgenics 2, as perhaps there are differences, different calcium microdomains which are driving this.
Yeah, great question. Short answer is we haven't looked. You know, the in vivo data we have suggests that, you know, we are limiting at least the downstream effects, whether I don't think we have actually done, you know, Western blotting or anything to look at just whether or not the initial stimulus or the initial signaling downstream of the iso stress is intact, but it's a great point.
Did you look at the levels of the receptors at all? Or is there
no, we
limit the
haven't.
antibodies? Okay. Okay, so second question is from Brian O'Rourke, and it reads, the key factor explaining differences between our studies is which calcium domain you are in, that is, in decompensated heart failure with depleted SR calcium load and poor mitocalcium loading. It is beneficial to enhance mitocalcium signaling to a normal level, not super normal level. However, in hypertrophic states with high SR calcium load, that is isoproterenol stimulated, increasing mitocalcium is probably a bad idea. Have you actually measured mitocalcium signaling during myocyte stimulation in your models to see if you are in a normal range or outside of it?
Yep, great question. So with our in vivo models, it's difficult for us to do. Again, in these chronic stress states, you know, when do you look? Do you look at week 1, week 2, week 3, week 4? We have some evidence that there's, even if we say overexpress NCLX, there's going to be some compensatory upregulation of the MCU. So the system will try to correct itself very rapidly. We've not yet done like isolated cardiomyocytes after these stressors and then tried pacing them. And the closest we've gotten is doing the neonatal rat ventricular cardiomyocytes with just acute pharmacological stimulation in vitro, but it's a great question. Okay,
so the next question is from Kim Helgren. Thank you. I enjoyed this talk. I may have missed this during the presentation, but how does NCLX overexpression affect ATP production in acute short-term stress, such as exercise or fight or flight?
Again, good question. So we don't really see any deleterious effects or any limiting effect of NCLX overexpression in our model. And I'll point out that with our model, it's not a very dramatic NCLX overexpression. It's maybe twofold at most. And a key point here is that the model is that NCLX-dependent calcium efflux is occurring more slowly than mitochondrial calcium accumulation through MCU. So in these acute stress states, we're probably still able to boost mitochondrial calcium enough to get these physiological signaling responses to support the ATP production, exercise, things like that. And then, maybe there's some low-level reduction of that signal on the back end through overexpressing NCLX, but we've not yet seen any sort of limiting effects.
So the next question is from Julia Liu. Great talk. I completely agree about the multifarious roles of mitochondrial calcium. Along the lines of Brian's question, to what degree does mitochondrial calcium change among your different hypertrophy models, TAC, angiotensin, PE, etc.?
Great question, Julia. So we've actually been limited here by some of the technological challenges, which I'm sure you're well aware of. So we always do the classic easy thing, which is to take your cardiac tissue at the end of your study, do a Western blot for PDH phosphorylation. But we also know from everything that's in the literature that over time, PDH phosphorylation state is going to be counter-regulated by activation of other signaling pathways. It's not a pure indicator of mitochondrial calcium content. So if we can do that, we can see some changes that suggest that we are increasing mitocalcium in our MCU transgenic models, and then preventing mitocalcium accumulation when we overexpress NCLX, but putting specific numbers on that has been very difficult. I mean, I think there's sort of a debate in the literature over whether you take intact tissue, isolate a cardiac mitochondria. By the time you're able to say lyse that mitochondria and actually measure the calcium content, did it really change from what was the in vivo state? So it's been difficult for us to do.
Okay, thank you. So the next question is from Moni Nader. Excellent presentation. In failing hearts, one of the therapeutic intervention is the administration subsequently, which indirectly slows down the NCX at the sarcolemmal to improve cardiomyocyte contraction. What is the effect of, okay, there are a lot of typos in this question. Okay, what is the effect of administrating subsequently on mitochondrial NCX and subsequently mitochondrial function?
Yes, I think the question here is getting at what effect will digitalis have on NCLX and the mitochondria.
Okay.
Okay. So I'm not aware of any studies that suggest that digitalis can affect NCLX activity. So again, this is a different exchanger than the plasma membrane sodium calcium exchangers, encoded by a different gene, SLC8B1. So it's very similar, it's the same family, but it's distinct. The classic inhibitor for NCLX that everyone uses, which again isn't all that great in and of itself, is a compound called CGP37157. But yeah, I'm not aware of anything that's been published so far suggesting that digitalis would or would not have an effect on mitochondrial NCLX, but it's an interesting idea.
Thanks for clarifying, because I completely messed up that question. Okay, let's see here. We still have a few more minutes and I don't see any more questions. So then I will end with one final question. So I was really intrigued by the data that you got from the MCU transgenic cross with the cyclophilin D knockout mice and what you're finding on necrosis, that you actually saw an increase in necrotic cell death with the trypan blue uptake. So did you do some EM studies to actually look at the morphology of the mitochondria? Do they display the typical swollen morphology, or what do they look?
So we haven't done that yet. We would expect that they do. Again, if we put these MCU transgenic mitochondria in, in an isolated system, measure the absorbance, give them a bolus of calcium, they will swell more than a pre-control mitochondrion. Mm-hmm. So yeah, we're kind of puzzled about what's going on with this cyclophilin D knockout and why it's not protective. one idea that we're trying to follow up a little bit is, we probably have excessive ROS production in these hearts, and that in and of itself could be enough to, say, oxidize membrane lipids, either within the mitochondrial membranes themselves to initiate some necrotic signaling pathways, or maybe even at the level of the plasma membrane to compromise plasma membrane integrity, and then give us that same sort of necrotic phenotype, but via different mechanisms than classical necrotic signaling.
Okay.
There's also some evidence from Tish Murphy's group suggesting that with MCU knockout, there can be sensitization of cyclophilin D to allow for increased necrotic cell death in response to ischemic injury in, say, a global MCU knockout model. So manipulating MCU protein availability and MCU-dependent flux over time seems to remodel a lot of the cell death signaling pathways. Now, whether MCU transgenic overexpression would have sort of the converse effect remains to be determined. If that would maybe desensitize cyclophilin D such that deleting cyclophilin D doesn't have any net effect, but you have upregulation of other death pathways that are still sufficient to kill the cells.
Okay. Okay. So now we have one more question, or a few more questions for you from Aurora Murakami. Amazing talk. I was wondering whether you checked mitochondrial morphology and mitochondrial shaping proteins levels in your MCU overexpressing and NCLX overexpressing models.
Great question. No, we haven't done EM on those.
Okay, and then we have a follow-up question from Moni Nader. Digitalis inhibits the sodium potassium ATPase, sodium overload inhibits the sarcolemmal NCX. Does this affect mitochondrial NCX?
So with respect to sodium, so this is, again, work out of Brian O'Rourke's group. Rather than inhibiting mitochondrial NCLX, there's data from their group suggesting that, it's in say end-stage heart failure situations where you have increased cytosolic sodium, that's actually going to drive increased flux through the transporter. So I don't know if that's the level of NCLX activity per se, but just the fact that you have more of this ion available to drive flux will result in a net increase in NCLX activity.
Okay, and now we have one more question. It's a good discussion here from Heinrich Tagmeyer. Fantastic. Was there any change in the coupling of glucose uptake and oxidation in the NCLX overexpressors?
Great question. We haven't checked glucose uptake.
Okay. And I think that's it for the questions. So,
all right, thank you all.
Good discussion here. So thank you again, Joanne, for a fantastic presentation and a good discussion after. And thank you everyone for tuning in.